Corinne, can you see it?
Yeah, I can see the PowerPoint.
Okay, I think we're all set then. Thank you so much.
Yeah, no problem.
Appreciate it.
See it?
Can you still see it?
I can still see it.
Okay, perfect. Okay,
Awesome.
well, thanks everybody for being here.
All right, good luck, Lexi.
Thank you. Does this record it?
It's recording it too. Yeah. Luckily, with our EDD program, I've done this like 1,000 times. Thanks everybody for being here. Lexi, look forward to your presentation. Lexi has been working to make it shorter, but it's still about 30 minutes long. But that way, it'll probably shorten our questions. She'll tell us everything we need to know. So the way I usually do proposals is Lexi, you'll present, and then if you guys have questions, you can start with questions. Then I'll turn it over to the committee for questions. And then at some point when we've exhausted our questions, we'll ask all of you guys to step out so that we can just decide what changes have to be made and things like that. And then I'll call you back in. All right, so Lexi, off you go.
Okay. Oh. Okay, thank you. Today I will be proposing my study entitled Exercise and Executive Function in Older Adults, Exploring the Mechanisms. So we know that the population of older adults in America and worldwide is increasing. In year 1930 in America, 2 out of every 100 adults was over the age of 65, whereas in 2030, it's expected that that is going to increase tenfold to account for about 1/5 of our population. And we should be concerned about this great increase in percentage of older adults because aging is associated with declines in cognitive performance. Specifically, there are declines in executive function, in memory, and in global cognition. Previous studies have found that declines in executive function tend to precede memory and cognitive and global cognition decline, and executive function declines are also predictive of declines in other cognitive domains such as memory and global cognitive performance. So it may be of interest to really put focus on executive function to potentially have a downstream effect on other domains of cognitive performance as well. Declines in executive function are associated with health risk, chronic disease, reduced quality of life, and mortality, and therefore creates this issue for this 1/5 of our population that they're going to have declines in cognitive performance that are associated with these negative outcomes. But before we get into how we might intervene with that, let's just go over what executive function really is. Executive function is made up of 3 major constructs: working memory, set shifting, and inhibitory control. Although that just for the purpose of this, going over it quickly in this slide, I'll just operationally define these terms, but then later I'll actually give you some really some good examples of tasks for these functions. So for working memory, it's the ability to manipulate contents in your temporary or working memory. So basically, it's also considered updating of working memory. I've just shortened it to say working memory. But when you're remembering something and then you're given new information and you're updating that information to then basically manipulate it. Set shifting is a disengagement of an irrelevant mental set with the active engagement of a relevant mental set. So if you're given 2 sets of instructions, the ability to go back and forth between the 2 when it seems relevant. And inhibitory control, by definition, is suppressing a dominant response in favor of performing a subdominant response. So with a health behavior example of this that's talked about in literature is if the goal is for to improve health, then you would want to inhibit reaching for that donut or eating the donut and in turn eat carrot sticks or something like that. So you're suppressing that more automatic response because you really want the donut because the carrot sticks are more associated with your goal that's in mind. And although aging is associated with and then these 3 factors also load onto a common executive function factor. So Miyake et al. 2012 showed showed this, that they all load onto this latent factor. So even though some studies use one of these factors and some studies use all, it's important to keep in mind that they do share some variance across, that they fall onto this common executive function factor, even though some of them, like working memory and step shifting, do also have their unique variance. So we know that executive function is going to decline in older adulthood, but similar to other domains of cognitive performance, such as global cognitive performance, this is not consistent across every individual. Every 70-year-old, every 75-year-old does not look the same. So there's this great variability in the age-related changes in brain structure, function, and cognitive performance that are largely due to biological, psychological, environmental, and lifestyle behaviors. So these individual differences can lead to differential outcomes as far as cognitive performance as well as brain structure and function. The cognitive reserve theory helps to provide some understanding as to the individual differences in these age-related declines. Stern speaks about the neural reserves and cognitive reserves separately. Neural reserves being talking about the actual structure, so the neurons and the synapses that allow for cognitive processing. So these are the actual structures, where cognitive reserves are really the ability to cope with the insult. So this is where we're talking about brain function. And even though they might seem to go hand in hand, they sometimes are this nice little kind of pull in between. So if you see, ooh, okay.
Be careful not to touch it like that.
Yes. So this group up here is not declining. So this is just global cognitive function. But when we think about the variability in age-related declines, I thought this really demonstrates that nicely. But just because someone has not declined does not mean their neural reserves have not declined at all. They could have a shrinking prefrontal cortex. They could have differential hippocampal volume, but they might be able to compensate with enough cognitive reserves. So if their cognitive reserves are kind of bolstering that effect, it would appear that there's no decline. But if we really look into it, they may be declining in different ways. It's just not apparent in behavior. But similarly, someone with moderate decline might have the same amount of neural reserves as someone with no decline, but a reduced cognitive reserve. So if they're not able to cope with these insults, if they're not able to compensate for these differences, then they are going to be showing to decline in behavior. It's just showing that higher reserves are allowing for resiliency in age-related declines really until a threshold is met. So even though someone has higher reserves, it's not that this is going to go forever. They're going to reach some critical threshold and then they would decline. But the idea is that that would be much later in life than someone with lower reserves. So we know that there's variability in the age-related declines. And we know that executive function is declining in older adulthood. So we might want to focus on interventions that have been shown to affect neural reserves, so affecting the brain structure, cognitive reserves affecting brain function, and cognitive performance. And of no surprise, exercise has been one of those interventions. Exercise has been shown to improve executive function in older adults in both the acute setting and also in response or in adaptations to chronic exercise. So these meta-analyses are showing that in older adults specifically, there is a moderate size benefit for acute exercise on executive function and a small and positive benefit for exercise on executive function in a chronic setting.
What's that effect size on the left, Lexy?
It is, for the older adults, is 0.67.
Oh, I see.
And then for the chronic, it's 0.34. So this is where the evidence comes out. Exercise improves executive function, but actually what's shown is that there's high variability in these designs and findings. Not every study shows this benefit, and a lot of people attribute that to individual differences. They attribute it to variability in the study design, such as exercise duration or intensity, and then the findings, what they're actually studying. So as I mentioned, some people choose to study one construct of executive function and others choose to study others.
That's okay.
Should I continue?
Just in time.
Just
in time. Hang on just 1 second. We'll let her get settled for just a second.
Really, really sorry.
That's okay.
It's okay.
I just only half an hour.
Lexy's really just on the background part, so we held out a few minutes, hoping it was just a short traffic delay.
Sorry.
That's okay.
That's okay.
But, so this is really sort of the, pardon me?
Do you want to turn off this?
It doesn't matter. We can go ahead and it won't hurt anything.
Okay.
But, so really she was just sort of doing lots of people, individual variation in cognitive form.
Okay.
And then now she's to the part that's really important, that is acute exercise, like 6-7 effect size for older adults, chronic exercise.
Okay.
All right?
Thanks.
Okay.
Go ahead.
All right. So as I was saying, there's high variability in the study designs as well as the findings. And one of these reasons might be that there's still a lack of mechanistic support. So there's still a large gap in the literature as far as specific mechanisms that explain how exercise affects executive function in older adults, and targeting different exercise intensities and durations and things like that might be kind of premature before we actually know how exercise is affecting executive function, trying to target those benefits. So to approach the gap, I feel that it's important to understand the acute exercise response to further gain an insight into the chronic exercise adaptation. And to do that, it would be important to focus on factors that respond to acute exercise, that adapt to chronic exercise chronic exercise programs, and that are related to improved executive function. Two of these factors are increased brain-derived neurotrophic factor, or BDNF, and changes in neural activation, or the blood oxygen level dependent signal or response, as measured by functional MRI. But for the purpose of this presentation, I will just refer to it as neural activation. So brain-derived neurotrophic factor, or BDNF, aids development, maintenance, and plasticity of our nervous systems, and it's typically decreased in older age. It also decreases in response to different disease states, so it's would be negatively correlated with health status, and decreased BDNF is associated with decreased cognitive function. Importantly, in a recent meta-analysis, it was shown that BDNF increases in response to acute exercise in humans, and this has been shown in much of the rodent literature as well. So Danoff et al. 2017 found that there was a moderate-sized effect on BDNF in response to acute exercise. And importantly, these increases are sometimes associated with the transient improvement in executive function. I say sometimes because in the young adult literature, there is some evidence to the contrary, but with older adults, there's only been one study, and that study has shown a positive outcome. So here we're looking at executive function tasks, and as this is really time, so as time's going down, the change in BDNF is going up. So this is a good expected association if we were looking at this. And then in this recent publication with older adults, we see that the post-intervention level of BDNF was associated with working memory.
So when you say it's time going down, you're saying increase in response time?
Increased interference, right? Because it's a delta MTB.
Okay.
Yes.
But yeah, faster response time.
Sorry, faster response time.
Just check.
Okay. Interestingly, when people are performing working memory, shifting, and inhibition tasks, there's a common network of brain regions that are activated during this that is associated with performance. These include regions such as the prefrontal cortex, the frontal polar cortex, the orbitofrontal cortex, and the anterior cingulate. And this has been termed the cognitive control network. This network also has some other terms to it, but for here, I'll just speak to it as the cognitive control network. Changes in this network and in structural integrity and functional connectivity can affect executive function performance. So those changes are associated with aging, and that is then downstream has negative effects on executive function. There's been limited research using neuroimaging to look at this interaction between neural activation and executive function. But in one FNIRS, or functional near infrared spectroscopy study, it was shown that older adults following exercise had greater activation in the right frontal polar region and that was associated with their Stroop interference or inhibitory control performance. So this was kind of one of the first studies showing this as far as using neuroimaging in response to exercise. There has not been any fMRI studies with older adults looking at this, but there have been fMRI studies looking at neural activation in response to exercise in other populations, such as this study with children showing differences in exercise, post-exercise compared to post-rest. In this study, looking at post-exercise, comparing bipolar disorder and healthy control adolescents. I don't think I said this, but the first study was in children. And then in this last study, looking at the same pattern in young adults. So again, looking at after exercise compared to after rest. But similarly, that we see a variability in age-related changes, we also see a variability in the response to exercise. So the response of BDNF the response of BDNF to a bout of exercise has been shown to differentiate between different populations. One study showed that depressed individuals actually had a greater impact or a greater response compared to healthy controls, even though they also started lower, so it was considered more of a normalization. And then in this study where I showed before, that overall post-intervention BDNF was associated with working memory. But when they stratified by baseline BDNF, they found that in tertiles, the lower levels of BDNF, actually their response was associated with the response of working memory. So this may suggest that those that may have lower cognitive reserves or in this lower BDNF state may be more reliant on certain changes or responses to have that response in executive function, where those with higher BDNF may not be as reliant on that response. The cognitive reserve theory may provide a framework, but it is missing key components. It does not typically speak to overall health, fitness level, or genetics of the individuals. This variability in the response to exercise suggests that there's a need for a more robust moderator to account for these individual factors to better understand this relationship between exercise and executive function. Where I'm proposing that vagal tone may just be that moderator. Vagal tone is a measure of parasympathetic nervous system input and is a proxy for the integration of brain regions that guide self-regulation, which both include executive function as well as emotional regulation. Vagal tone decreases with aging and disease, is influenced by individual factors such as genetics, fitness, and environmental factors, and is associated with executive function in young and older adults. Importantly, in terms of this proposal, it interacts with BDNF in humans and rodents. Here I'm just showing the interaction of in humans, where we're looking at different BDNF genotypes. The Val genotype is associated with greater expression of BDNF as compared to the Met genotype. And here they also saw that in the Val group, so that high expression of BDNF group had greater vagal tone or high frequency power, which is a proxy measure of vagal tone, as compared to the low BDNF group. There's probably more specific mechanistic support in rodents from previous authors like Wan and Griffian, but for the purpose of this presentation, I'll just leave it at that. Importantly, vagal tone is controlled by this autonomic control network. Thayer has described this in great lengths, him and their colleagues in the Thayer papers in Smith et al. 2017, showing that higher vagal tone is associated with higher functional integrity of these control networks. So Sakaki et al. 2016 furthered that by looking at the connectivity of the medial prefrontal cortex and the right amygdala, which are regions that are both associated with cognitive control and emotional regulation, and found that the connectivity between those regions is associated with another heart rate variability proxy measure of vagal tone in both young and in older adults. So this is important, and this actually provides support for the neurovisceral integration theory as well, suggesting that from a top-down perspective, the medial prefrontal cortex and the amygdala, their connectivity is associated with vagal tone. So the purpose here of this study is to look at these relationships. So I am suggesting that there's preliminary evidence to suggest that acute exercise, or there's great evidence to suggest that acute exercise is going to benefit executive function in older adults. And these may be mediated through factors of BDNF and neural activation. But the magnitude of increase of BDNF and the magnitude of change in neural activation may depend on resting vagal tone. So it may be influenced by kind of that whole system perspective rather than what some people have done in the past, looking at very specific individual differences. And although this looks like a very exciting moderated mediation model, here we will not be testing that per se because there is not enough preliminary evidence available to suggest that all of these relationships will hold. So what's important for this proposal is to look at these independent relationships to then inform a much larger study that would be powered to test this moderated mediation model. So I've gone over those aims. Okay. So to test our aims, our participants will be 20 healthy right-handed older adults, 65 to 80 years old. This will be a within subjects counterbalanced by conditioned design with morning sessions only that are spaced at least 1 week, but no more than 2 weeks apart. Just to briefly go over the entire design, participants will come in and be consented for the entire study and be screened for everything they will be participating on days 1 and days 2 and 3, just to ensure that they'll be able to be included. They will complete questionnaires specific to their demographics, and then I'll take their blood pressure, height, weight, and resting heart rate. They will then complete baseline executive function tests, the baseline blood draw, and complete a submaximal exercise test. This will all happen here in this building in the physical activity and cognition lab and the exercise physiology lab. On days 2 and 3, they will meet over at the Joint School of Nanoscience and Nanoengineering, again be screened for safety concerns, have vision tests to ensure that they will be able to see the font on the screen, basically, just to make sure they can see the tasks while they're in the scanner. They will have a resting state fMRI and a structural MRI prior to anything that they do that day. Part of this in this proposal is to habituate them to the environment. I will not actually be analyzing that data for this proposal, but given that once they finish their exercise or resting bouts, they're going straight into the scanner and starting the tasks, we felt it was an important part that they habituate to the environment before they start these tasks. After those baseline MRI and resting state fMRI, they will have a preconditioned blood draw, either exercise or rest, and then and then have a post-conditioned blood draw, and then immediately go into the scanner and complete the executive function tests, the same test that they did at baseline. So they'll do the same ones each day. So getting a little bit more specific in some of the methods, resting heart rate is really, we're really looking at heart rate variability here. So we're assessing resting heart rate through R-to-R intervals to look at heart rate variability that can be reduced into vagal tone. We'll this will be measured by the Polar V800 chest straps, and it will be measured for 8 minutes. Out of those 8 minutes, when reducing the data in Kubios, we will find a 5-minute window after inspection for artifacts, a 5-minute continuous window of data to then reduce into high frequency power. Although from Kubios, we will get all of the heart rate variability metrics, specific again to this proposal, we'll only be looking at high frequency power. For executive function, we'll measure inhibitory control, set switching, and working memory in that order. For inhibitory control, we will use the Stroop color word test. In case one of you really isn't familiar, but I think you are anyway, I'll go over it anyway. So the Stroop test has 3 conditions that we'll have: the congruent, incongruent, and neutral, and then interspersed with resting blocks. So you'll see the resting blocks over there where the participants are just staring at a fixation. For the congruent, for all of the tasks, the participants are told to respond to the ink of the words or the non-word letters. So for congruent, super easy, red is in red ink, blue is in blue ink, but for incongruent, red is in blue ink, blue is in green ink. So it gets a little bit more challenging, and then for neutral, they're just responding to the colors of the X's. These tasks are designed to be scanner friendly in a block design, but they will be the exact same at baseline. So even though it's not necessarily needed from a computer point of view, we did it to keep the task the exact same. The measures here that we'll look at is the reaction time during correct response trials and % accuracy, and inhibition will be calculated by incongruent minus neutral mean reaction time.
Lexi, before you go on, I'm sorry.
That's fine.
So the way you show it there is like the trials are alternating.
The trials they were counterbalanced.
So how do they know if it's a congruent trial or an incongruent trial? Oh, I guess
They don't.
Right, it doesn't matter because they just always have the same directions. And so it's counterbalanced and randomized.
Yeah.
Okay, I'm with you. I just wasn't thinking. What else?
For set switching, we're using a category switching task. This task was originally developed by the Engel Lab in Georgia and has since been just modified slightly. And the way that it's designed has also been used in the scanner before. The H blocks are a high switching load block, where the L blocks are a low switching load block. The high switching load is 70% of the trials will be switching, where the low is 20% of the trials will be switching. So you get an idea of what it looks like. Participants are told 2 sets of instructions. If you see a heart, you're deciding whether the word underneath of it is alive or not alive. If you see a soccer ball, you're deciding if the word is something that's smaller or larger than a soccer ball. And so in a repeat trial, they're seeing the same cue back to back. So they see the heart with a lion, alive or not alive, a heart and a pebble, alive or not alive. And then in a switch trial, they see the heart, pebble is not alive, and then with the shark, or they see the soccer ball, shark is larger. So they're having to switch between the relevant information. Switch cost will be assessed for accuracy and reaction time by the difference of switch trials minus repeat trials. And then lastly, for working memory, we will be using the plus task. This has been a task that's been used in the scanner before, as well as a task that we've used in our lab during our sleep deprivation study, where participants are seeing 2 letters separated by a fixation, and they're told to shift those letters forward 1 spot in their mind and then respond to the probe either correct or incorrect. So if they see a B and a J, after a short delay, if a C comes up or if a K came up, it would be correct. If any other letter came up, it would be incorrect. Working memory performance will be assessed by accuracy and reaction time during the correct trials. For the blood draws, they'll have 5 blood draws, pre and post exercise, pre and post rest, and then the initial one at baseline. Each blood draw will be 20 milliliters of blood, and the processing will be identical to what was suggested by Pollicini et al. 2015. This article really just went over the best method to process blood when wanting to quantify BDNF. So as such, we'll use that here. Then we will use an ELISA from Avicera Bioscience, which was suggested in that paper to be the best out of the available ELISAs to assess BDNF, and it will be for mature BDNF specifically, and this will be quantified in picograms per milliliter. For the submaximal exercise test, we'll use the Ekblom-Bach cyclergometer test. I decided to go over this test just because it's not that commonly used. It's been recently validated in older adults, and it's also shown to estimate VO2 max at more accurate than Astran rhyming, which has also not been validated on older adults. So for those reasons, I chose to use this as the submaximal exercise test. It's 2 stages, 4 minutes each. In the first stage, every participant is cycling at 60 revolutions per minute against 30 watts. And then in stage 2, for 4 minutes, they self-select watts based on an explanation of what a rating of perceived exertion of 14 and 15 is. And they select that and still maintain that 60 revolutions per minute. And this is used to predict VO2 max to then prescribe the exercise intensity. These equations were the equations that were recently validated in the Bjorkman paper. And I won't really go over that, but what you are putting into it is the change in the heart rate and the change in the power output from stage 1 to stage 2. From that, we will calculate VO2 reserve to get the correct prescription range for 55 to 65%. And then from there, we will transfer that into prescribed watts. So it will be 55 to 65% VO2 reserve and as prescribed through a specific watt range. They will be exercising on the exercise condition day for 30 minutes. 5 of those, the first 5 minutes will be more of a warm up, and then 25 minutes of exercise on a cycle ergometer. This is just a picture of a previous study where our young adult was exercising in the scanning suite, so you could get a feel for what the environment looks like. The rest condition will be the exact same to the exercise, other than the fact that they will just be resting on the bike, not cycling. Both conditions will be exposed to an educational video, and the measures will be, watts will be recorded every minute, heart rate will be recorded every other minute, and rating of perceived exertion every 5 minutes. To test our specific aims, initial preliminary analyses will use a repeated measures MANOVA to assess the effect of condition on measures of inhibition, switch cost, and working memory. Then to start getting into the aims, to our first analysis will include a paired samples t-test to test the effect of condition on change in BDNF. Then we'll use a linear regression with condition, vagal tone, and an interaction term of condition by vagal tone to assess the influence of vagal tone on the magnitude of change in BDNF. If number 2 is significant, then we will do a follow-up regression that includes condition, BDNF, an interaction term of condition by BDNF, and use vagal tone as a covariate to predict change in executive function. And if it's not significant, we will run the same regression, but we will remove vagal tone from the model. To test aim 2, we will use a 1 samples t-test on the change contrast from post-test to compared to post-exercise. So for each individual, we will have a comparison of the parameter estimates from post-rest to post-exercise. And then that will be entered into a 1 samples t-test to see if that change is different than 0. Further, linear regression will test the relationship between change in neural activation and vagal tone. If that's significant, then vagal tone will be used as a covariate to assess the relationship of change in neural activation with change in executive function. And if not, the follow-up model will be run without vagal tone as a covariate. I didn't know that was so hard to see. I could have put a background, but for our pilot data, we just had 2 older adults go through the protocol, everything except for the blood draws. And what we found was that it does appear that some of the things look as expected and some maybe not as much. It does appear that the pilot subject 1, who's in blue, had a bit of a speed accuracy trade-off, which could be expected, which is why we always look at accuracy and reaction time without leaving one of the 2 out. And then the subject 2 looked to be kind of going in the right direction for everything we expected. Importantly, for the fMRI analyses, there was significant clusters of activation that were greater for post-rest compared to post-exercise. And these were in the anterior cingulate cortex, the medial prefrontal cortex, the superior temporal gyrus, and the occipital and frontal polar areas. And so these are areas that are included in the cognitive control network. So that was a positive finding. Those were not necessarily, they were more analyzed as 4 separate people rather than 2 people within subjects, because you can't analyze 2 people, or I couldn't. I don't know if you can. I don't think you'd want to. So even though there's not enough there's not anything in the literature that would suggest that whether exercise would lead to compensation or greater activation or more efficiency or reduced activation with better performance. This out of pilot stuff may suggest that it's more efficient, seeing that there was nothing significant for post-exercise compared to post-rest, only when we compared post-rest over post-exercise. So just to conclude here, we expect to find preliminary evidence of some underlying mechanisms involved in acute exercise and executive function, including the factors of vagal tone, BDNF, and neural activation. We think this will help better understand the acute session of exercise, of how exercise can affect executive function, and be the first step of how to look at how exercise cumulatively affects executive function. The next step, as I mentioned in the beginning, would to test this design on a much larger scale to really look at a moderated mediation model. And then the long-term goal would be to design individualized exercise prescriptions to have the greatest effect on executive function, with the potential to have a great impact on health risk, chronic disease, and mortality in older adults. So just a quick thank you to my committee and to my research assistants who aren't here, but I really appreciate them, so they're there.
Thanks, Lexi. Right, Lexi, come sit down. Travis and Jared, if you guys have questions, I'm gonna let you guys go first, but I'm gonna stop this recording. Stop our YouTube.
That was great. I have nothing.
Thanks.
I have a couple.